Hello everyone, my name is Oluwaseun Inshallah. With me is my colleague, Ms. Vijita Rasali. Today, we will be giving a talk on labeled quantitation. Our paper of interest is titled Proteome Dynamics of Developing Acaí Berry Pericarp, and this paper was written by Moab Andrade and his colleagues from the University of Ceará, Fortaleza, Brazil. Paper was published on the 9th of December 2019. This paper explains the quantitative proteomic analysis of the developing Acaí pericarp. Further, it gives information about the protein deposition pattern with regard to synthesis of amino acids, carbohydrates, fatty acids, and secondary metabolites. Also, it can be a model based on which the biosynthetic pathways of the secondary metabolites for the Acaceae family can be studied.
The acai palm is widely distributed in the Amazon basin, where the rich extract from the pericarp is a traditional staple in the diet of riverside and the urban population. The perceived benefits of such a berry as a health supplement expanded its consumption beyond the Amazon region and is currently in high demand all over Brazil and abroad. This ever increasing demand reached such proportions that the amount of fruits produced via extractivism is 10 times smaller than that of intensive farming. The limited knowledge about the biology and the biochemistry of this crop hinders its genetic improvement. Also, no commercial significance of these seeds have been established, resulting in ecological seed and disposal issues. The acai pericarp is rich in fatty acids, proteins, and dietary fibers, but it's mainly attributed to anthocyanins. The predominance of two anthocyanins, cyanidin 3 rutinoside and cyanidin 3 glycoside, have been observed. Also, flavonoids, homoerythrin, lignans, and tannins are also documented. During fruit developmental stage, the biosynthetic pathways yield these secondary metabolites. Therefore, from a plant breeding point of view, it is very important to study the dynamic changes of the developing pericarp of acai berry proteome, which will allow access to the protein pattern and pathway regulation. Moving on to the methods. First, sample collection. The berry fruits were collected from naturally growing plants in Ceará, Brazil, weighing between 0.5 to 1 kg. Collected berries were grouped into 12 developmental stages according to the maturity scale as shown in figure 2. Then they were further classified based on morphology into developmental stages as S1, S2, S3 and S4, and each developmental stage were in 3 biological replicates. S1 is associated with cell division, S2 with cell expansion, S3 is the last stage before maturation, and S4 is the fully ripe fruit.
Now to protein extraction. The collected acai berry samples were frozen with liquid nitrogen. After lyophilization, the pericarp were carefully separated from seeds and ground. Lipids and pigments were removed by immersion in pure acetone, then dried at room temperature. The samples were further ground to reduce particle size. 40 mg of samples were homogenized with 80 microliters of extraction buffer at a pH of 8. A phenolic phase was carried out next and further extraction with the extraction buffer. Precipitation step was done with 0.1 molar ammonium acetate in ethanol, and lastly a centrifugation step was repeated with the pellets obtained, washed and dried at room temperature. Now we digest our protein samples. We solubilize our samples first, then we reduce them with 10 millimolar of tris-TCEP, a denaturating agent, followed by a calculation using 40 millimolar of iodoacetamide, which hinders disulfide bond formation. Digestion step using trypsin, and is stopped by 10% trifluoroacetic acid. We do a further cleaning with centrifugation and separation by porous R2 resin, which helps us elute the salts and concentration of the peptide. Now, to quantitate our samples, our choice of label is iTrack. Peptides were labeled with 4-plex reagent according to the manufacturer's specification. In the labeling, the reagents corresponding to different reporter ions of 114, 115, 116, and 117 were distributed to samples S1 down to S4, respectively, in triplicates. Hydrophilic liquid chromatography was used to fractionate samples and eliminate excess iTrack 4-plex reagent. The elution of peptides was monitored by means of absorbance readings at wavelengths of 216 and 280 nanometers. The collected fractions were grouped with care to separate the highest intensity peaks, resulting in 7 peptide fractions for each biological replicate and dried.
For the nano LC-ESI MSMS analysis, the pooled samples were analyzed in triplicates by nano LC coupled to a MS quadrupolo orbit trap. The samples were loaded in a C18 trap column and the peptides were separated by a C18 column. The ionization and transfer of peptides was done by using a nanoelectrospray source. The mass spectrometer was operated in data dependent analysis mode and the higher energy collision dissociation was normalized at 30 normalization collision energy. Chromatograms and the spectra were visualized by Excalibur 2.1 software, in which the chromatographic standard was checked for complexity of each fraction and reproducibility between biological replicates. For data analysis, the spectra obtained were analyzed using Proteome Discover 2.1 software. Identification of the peptide was carried out by peptide spectrum matching with sequenced HT algorithm against a protein database of 127 common mass spectrometric contaminants, which was present in MassQuant software package, and of the Acarce family along with Arabidopsis. False discovery rate was estimated by target decoy PSM validator tool that processes the data against a reverse database. Pattern Lab for Proteomics version 4.0 was also used for protein identification and quantification. It generated target decoy database to include a reversed version of each sequence found in the Acarce family combined with Arabidopsis, and those from 127 common mass spectrometry contaminants. Comet search engine was used for searching spectra against the target decoy database and to converge a list of identified proteins, which was less than 1% FDR. YADA was used to eliminate multiplex tandem mass spectra from the database. An isobaric analyzer module was used to extract the iTrack reporter ion intensities and then normalize the signal from each isobaric marker.
Now to the quantitative analysis of our Acaí proteome dynamics. A total of 4,286 proteins were identified, of which 476 had differential abundance. Looking at figure 3, we can see that the transition from S1 to S2, a total of 156 proteins decreased in relative abundance, while only 12 had its relative abundance increased. And a similar trend was found comparing S1 and S3. However, from S2, the pattern is reversed as development progresses, in the sense that the number of proteins displaying an increase in relative abundance steadily increased. The identified proteins were associated with various metabolisms of carbohydrates, lipids, and amino acids. Diverse sets of proteins involved in secondary metabolites synthesis were also identified. 77 proteins related to carbohydrate metabolism were differentially regulated in developing Pericarp samples, out of which 44 of them were upregulated and 14 were downregulated, but only 23 did not develop or show a regular pattern of synthesis. On this figure, to consider carbohydrate metabolism, looking at figure 4, the decrease in relative abundance of fructose bisphosphate aldolase with a fold of 0.5 indicates the likelihood that the glycolytic activity was higher in S1 compared to S2. Also on the same figure, enzyme pyruvate dehydrogenase increased in differential abundance. Such data implies that the pericarp development and metabolism is geared towards the supply of energy for cell division, expansion, and formation, as in the early developmental stages, while at late stages, S3 and S4, metabolism is directed towards the accumulation of secondary metabolites and nutrients, leading to food maturation. For the secondary metabolism, in the course of pericarp development, 281 proteins were identified as involved in secondary metabolism. From this total, 56 were differentially abundant. Out of the 56, 23 were involved in phenylpropanoid biosynthetic pathway. This pathway is relevant as it provides metabolites which have the adaptive role of conferring defenses against biotic and abiotic stress. This pathway is of use to the acai berry pericarp as it provides precursors for biosynthesis of flavonoids. An important enzyme of this pathway is phenylalanine ammonilyase, PAL, with a relative abundance at S3 as shown in figure 5 and 10 times greater in S4. Other enzymes related to this biosynthetic pathway of phenylpropanoids are shown on figure 5. The list of data obtained overall tells us that this pathway is active during the development and in full intensity at stage 4, which is the fruit development stage. The higher level of PAL in S1 and S2 compared to S3 shows that the phenylpropanoid pathway is orientated towards the production of defensive metabolites and the production of lignified cell walls, which are typical in the early stages of development, as in S1 and S2. 4 key proteins also involved in the biosynthetic pathways of flavonoids and anthocyanins were also identified. Anthocyanidins, 3-O-glycosyltransferase, 3GT, flavonoid 3'5' hydrolase, leucoanthocyanidin dioxygenase, and UDP glycosyltransferase 89B1, all displayed higher relative abundance in S3 and S4. F3'5'H is involved in the biosynthesis of delphinidine-based anthocyanins. LDOX catalyzes the oxidation of leucoanthocyanidins into anthocyanidins. 3GT uses the UDP-D glucose to glycosylate autocyanidins, giving rise to anthocyanins. UGT89B1 is an enzyme that acts as a regulator in homeostasis of phenylpropanoids and flavonoids, hereby modifying the solubility, stability, and toxic potential of this compound. The LDOX and 3GT at the last developing stages of S3 and S4 is also associated and evident in the color change and anthocyanin accumulation in the acai berry pericarp.
The last stage of pericarp development and the relative abundance of enzymes from biosynthetic pathways other than the phenylpropanide pathway were identified, and its protein deposition pattern were determined as shown in figure 7, thus indicating the presence of relevant metabolites in the mature pericarp. It is interesting to note that these metabolites have pharmacological and nutraceutical potentials, as it is in the case of pterostabilin and furaniol. Up to now, the presence of these substances in the acai berry has not been experimentally verified. In this study, we quantitatively analyzed the proteome dynamics of fruit pericarp of the acai palm. In total, we identified 4,286 proteins, and from these, we determined the differential abundance of 476, most of them involved in the biosynthetic pathways of carbohydrates and secondary metabolites. The analysis revealed that the enzymes involved in metabolism of carbohydrates and in the phenylpropanide pathway are more abundant in the early and the late developmental stages, so as to meet the metabolic demands for cell division, lignification, accumulation of phenolics, and for the metabolic changes related to fruit metabolism. In the late stages of pericarp development, increased relative abundance of pterostabilin and furaniol compounds was observed with pharmacological and nutraceutical properties. The future direction of this study should focus on deep metabolomic analysis of the 4 developmental stages to uncover the full pharmacological and nutritional properties. Also, these studies will pave the way for studies aimed at the genetic improvement of the nutritional properties of this fruit crop and to the use of seeds as a source of raw material for the production of bioenergy. These are our references. Thank you.